Good evening, Pratham. Okay. So today we'll be carrying on with our course, and then in the next class again we'll be having a Zoom class for the discussion. Okay then. So today we'll be proceeding our course with the electron transport chain. Okay, which is the pathway that comes after the Krebs cycle. Okay. So I'm Rahul Saha, and today we'll be talking about the electron transport chain. Like electron transport chain, basically what is electron transport chain? Like why do we eat food in the first place? So we eat food to get electrons, directly to get electrons, nothing else. The basic role or the basic mechanism or the basic fundamental role of any food particle that we eat is for us to get electrons, right? Without electrons, electron cannot flow in the mitochondria from the complex 1 to complex 4, and if it cannot flow, it cannot produce a gradient, which I'll show you right now. And if the gradient is not produced, we cannot form ATP. Simply what happens is when we eat food, okay, something known as electrons are shed out of the food, and this molecule is produced, NADH molecule, that is nicotinamide, adenine, dinucleotide, you can say hydrogen. Now this is a reducing equivalent. Okay, in the Krebs cycle we had seen that production of NADH, also production of FADH2. Okay, flavine, adenine, dinucleotide, then dihydrogen. So we had seen the production of these two reducing equivalents in our Krebs cycle only. Okay, in our Krebs cycle only. Now, if we have seen this in the Krebs cycle, what does it mean? Why does Krebs cycle occur? I have told you that Krebs cycle occurs mainly because not to produce one GTP which is equivalent to ATP. Krebs cycle mainly occurs in order to produce NADH and FADH2. So one NADH is produced from the pyruvate molecule when pyruvate is oxidized to acetyl CoA, plus CO2 comes out, oxidative decarboxylation process via the pyruvate dehydrogenase complex, which is E1 plus E2 plus E3, right? It's a huge macromolecular enzyme. Another 3 NADH is produced in the Krebs cycle, okay, from when you produce, you can say, from isocitrate, okay, to alpha ketoglutarate, okay. Also, then these kinds of events they produce Krebs cycle, okay. Then alpha ketoglutarate to succinyl CoA, again you produce NADH. The thing is, in total, when the two pyruvate molecules are produced in glycolysis, so two pyruvate molecules, when they completely go through the Krebs cycle, what we get is we get a total of, you can say, 3, 6, 7, 8. So 8 NADH and 2 FADH2. So we get a total of 10 reducing equivalents, means they are carrying electrons. They are these, simply I can say, electron carriers. Okay. Now what is the role of these electron carriers? These electron carriers, what they do is they will in turn let you produce the ATP in the inner mitochondrial membrane. So that is their main basic role. Okay. Now how do we comprehend it? Number one is NADH. Okay. When you see electron, right? When you see an electron, good evening, Samist. So when you see an electron, okay. So we have this mitochondria which we call as the energy producing organelle. Now why is it called an energy producing organelle? We'll talk about it, okay, in the electron transport chain theory. Okay. So I'll go really slow for you. Samesh, I have completed the errors in cell division chapter for you. Okay. So you can go through the lecture, and next class we'll be having on Zoom again on Saturday. So you can clarify your doubts and we can revise every doubt that both of you can have. And if you want some topic to be discussed, we can discuss that topic also in that class. Okay. So the thing is, this is the outer mitochondrial membrane, this is the inner mitochondrial membrane. Okay. Now, in the inner mitochondrial membrane, there are multiple, you can say, transmembrane proteins, multiple transmembrane proteins. So let's zoom in the inner mitochondrial membrane only. Let's draw only the inner mitochondrial membrane. This is the inner mitochondrial membrane and these are the transmembrane proteins. Okay. Present on the inner mitochondrial membrane. This is known as complex 1, this is known as complex 2, this is known as complex 3, this is known as complex 4, this is known as complex 5. This complex 5 is also known as F1 F0 ATPase. This is also the name of complex 5. Okay. Now what happens is NADH is the electron carrier which will donate its electron. One electron will be donated to complex 1. Like this 2 NADH molecule will come across. They'll donate their electron to complex 1. Now since a pair of electron, when a pair of electron passes through complex 1, now understand this process. Complex 1 is a better electron acceptor compared to your NADH. So it will accept electrons, okay, from NADH very spontaneously as per the thermodynamics. Okay, so they will get reduced, oxidizing NADH to NAD plus, right? So they will get reduced. Now they are accepting the electrons very easily. Okay, so the thing is, after a pair of electrons is accepted, okay, the simplest theory is 4H plus is pumped from the matrix to the, this is the intermembrane space, that is the space between the inner mitochondrial membrane and the outer mitochondrial membrane. Okay, now that is known as intermembrane space. Now 4H plus is thrown out of the entire, you can say, matrix. Now like this, fat H2 will also come. Fat H2 enters on the second complex, it will donate. Okay, same thing will happen. Okay, so here 2H plus is donated and 2H plus will go and form water, that will be seen how it is in complex 4. So the thing here is what, electron is being transferred from complex 1 to 2 to 3 to 4. Okay, means complex 4 is the best acceptor, followed by complex 3, followed by complex 2, followed by complex 1. So this is the best acceptor of electron. Every time 4H plus, and in case of complex for 2H plus, why, I'll explain it to you later on. H plus is thrown out of the membrane. Okay, like that. So what are you getting? What are you generating? Look at how the cell is so versatile. What are you generating and getting is, you are generating an electrochemical gradient, just like I had explained to you in case of neurons. Electrochemical gradient. So in the neurology action potential we were talking about, in case of neurology or neuroscience, we saw that the action potential, the electrochemical gradient of K plus differs from that of Na plus. Here also we are generating an electrochemical gradient. How? Look here. All the H plus present in the mitochondrion, the matrix of the mitochondrion inside the inner membrane, is being pumped into the intermembrane space, right? Since it has been pumped into the intermembrane space, what do you mean? That more and more H plus, okay, are now in the intermembrane space compared to the matrix or not, right? So more H plus, I can write it down, is in the intermembrane space compared to the matrix of mitochondrion. That is theory number 1. Theory number 2, more positive charge is in the intermembrane space compared to the matrix of the mitochondrion. Right or wrong? Because with each H plus, what I am doing is, you can see this diagram, with each H plus, I am throwing out a positive, you can say, ion also into the intermembrane space or not. That I am throwing out 2H plus means what? Here 2 positive ions, here 4 positive ions, again here 4 positive ions, here 4 positive ions. So this is becoming more and more positive, means automatically these charges have come from here. So here the increase of what will happen? Negative charges, because everywhere you have negative and positive ions both. So chlorines and the ion of chlorines, especially the proteins, will be present inside. So when you are throwing out positive ions, just like you throw out 3Na plus and take only 2K plus in through the NaK plus ATPase, you build up the deficiency of positivity, thereby you increase one negative charge each cycle of that specific ATPase. Same way, since you are throwing 4 positive outside, what you are doing here? You are generating the deficiency of positive charge inside the mitochondria or in the mitochondrial matrix, right? So what you are doing is, by generating the deficiency of positive charge, you are causing the excess of negativity or negative charge or not. So inside will be more negative, outside will be more positive, right? Outside will be more positive. So the outside being more positive, the inside of the matrix, or the matrix being more negative, the inside of the inner membrane, and the intermembrane space being positive. So I can say intermembrane space, the space between the outer and inner mitochondrial membrane, the intermembrane space being positive and the matrix being negative, is what? Is an electrical gradient or not? Is an electrical gradient. Then when 4H plus and 4H plus all the time in the complex for 2H plus, means H plus are being effluxed to the intermembrane space from the matrix. So H plus concentration is greater where? Where does the H plus concentration become greater? In the intermembrane space compared to the matrix. And we also know, you must have read this equation, that pH is equal to minus log H plus. So pH is directly proportional to the concentration of H plus that we know. More H plus, lower the pH, more the acidity. Lesser H plus, higher the pH, lower is the acidity, higher is the alkalinity or basicity, right? So where will we see more acidity? We will see more acidity in the intermembrane space, because we have here more H plus, so more acidic. That is, I can write down that in case of H plus, right? In case of H plus, more is present in the intermembrane space. Intermembrane space is highly acidic, thereby the pH will be lower. Suppose the pH is plus 2 and the pH of the matrix will be higher. Okay, because H plus will be lower there because H plus has been effluxed out. So pH of the matrix will be higher. Suppose pH of 5. So this is known as a chemical gradient. Now when we have produced an electrochemical gradient, what would happen is after a certain point in time when this entire NADH2 complex 4 cycle is done, enough H plus enough number of H plus have been moved to the, you can say, intermembrane space. What will happen is, by the law of nature, by the law of physics, you can say, they will come down their chemical gradient and try to equilibrate the chemical gradient and electrical gradient. So they will try to equilibrate or equilibrate their electrochemical gradient or not. So they'll come down means they'll come back into the matrix, so that electrical gradient and chemical gradient both can be equilibrated, both can be equated. When will that happen? That will happen through your complex 5 or F naught, F1, F naught ATPase. Means all the H pluses will come back, will return to the matrix via the F1, F naught ATPase transmembrane protein. And this transmembrane protein is a very, very special, very, very unique molecular machine which rotates every time. I'll show it to you later on. It will rotate. It is known as rotational catalysis. So it rotates 180 degree every time. Okay, one H plus goes through, and then due to that energy derived from the influx of the H plus from higher concentration to lower concentration means it is coming down its concentration gradient, means it is coming down its potential energy. So it comes down, some potential energy will be released, and that potential energy will be harnessed to cause the phosphorylation of ADP. Means ADP will be attached with the phosphate by harnessing the potential energy, because electrochemical gradient is high here, low here. It is coming down its electrochemical gradient. It harnesses the potential energy of the electrochemical gradient to attach phosphate group, free phosphate present in the intermembrane space in the matrix of the mitochondria, to attach with the ADP, adenosine diphosphate. So adenosine diphosphate will be phosphorylated to produce ATP molecule. That is how ATP synthesis is done in the mitochondria. That is the overview of this process. That is what is known as an electron transport chain and culminates at the F1, F naught ATPase or complex 5. So if electrons never got transported, if and only if electrons never got transported, what would have happened? Would you be generating ATP? Not at all. You would never ever be able to generate ATP. So why do we eat food? Vitamins, proteins and all fine, but biochemically, energetically speaking, we eat food in order to get electrons, in order to acquire electrons. That is the reason from now on you can tell your teacher, you can tell your friends that we eat food mainly to acquire electrons, so that our electron transport chain could be run properly, it could function properly, and then only will our complex 5 or F1, F naught ATPase will be able to harness the energy derived from the electrochemical gradient. When the H plus comes back, it returns into the matrix to generate ATP by causing phosphorylation of ADP to produce ATP. And this electrochemical gradient theory is known by the name as chemiosmotic theory. This theory only, the theory that I had explained to you, is known as chemiosmotic theory. Very, very important theory for your from your examination perspective. In your school examination, directly one thing can be asked: what is chemiosmotic theory? This is your chemiosmotic theory: build up of the electrochemical gradient, then the return, okay, of the H plus through the F1, F naught ATPase. That is known as the chemical, or sorry, the chemiosmotic theory, by which ADP can be phosphorylated to produce ATP. Okay, so this is known as your chemiosmotic theory. Also, one more thing I'll tell you, this is also known as oxidative phosphorylation. Now, why is it known as oxidative phosphorylation? Okay, that is again a very good question. Okay, it is known as oxidative phosphorylation. So, oxidative phosphorylation means what? You are oxidizing. Okay, are you not oxidizing? You just look here. NADH is transferring E minus or electron to the complex one. So NADH is getting oxidized or not, and this is getting reduced. So this is known as oxidation. NADH will become NAD plus. Okay, that is oxidation after the transfer of electron, and then when complex 1 will transfer to complex 2, complex 1, the intermediates of complex 1 will be oxidized, and so on. Right, when they will transfer, they're reducing the next partner and they're oxidizing themselves. That is oxidation. And this oxidation, continuous oxidation process does what? It performs what function or which function? It performs the function of generating this electrochemical gradient, and this electrochemical gradient, what it does? When the H plus comes back through the complex 5, through the F1 F0 ATPase, it is able to phosphorylate adenosine diphosphate to produce ATP. That is why it is known as oxidative phosphorylation, phosphorylation of ADP to produce ATP via these continuous oxidative processes. That is why it is also known as oxidative phosphorylation. Okay, is also known as, you can say, oxidative phosphorylation. So very important theory these are. Oxidative phosphorylation, okay, generation of chemiosmotic, or what is chemiosmotic theory? That is the generation of the electrochemical gradient. Okay, so in your book this has been given in a very complex way. Okay, concise and complex, will never be able to comprehend. So this is the real meaning of the chemiosmotic theory. Like whoever I encounter, they can never explain this theory to me completely, because they have directly mucked up from the book, and the book is very complex. It directly given oxidation, this happens, these are the carriers, everything happens, no nothing of the sort, no explanation, everything in a zest like manner. That becomes very hard for anyone to comprehend directly from the book. So this is how you should be knowing it through concept. Now I don't think you'll be able to forget this concept. Now you can write it in your own words in your school examination also, right? Now talking about the mitochondria. Okay, so the mitochondria, okay, we all are aware of the fact that the mitochondria are having two membranes. Okay, so the outer mitochondrial membrane is readily permeable to small molecules. Okay, very readily permeable to small molecules and ions. Okay, means which can move freely through the membrane channels. Okay, which are membrane channels are what? They are formed by integral membrane proteins. So always know for a fact that the outer mitochondrial membrane is readily permeable to ions and small molecules. Very important. Next thing is the inner mitochondrial membrane, on the surface of which, or on the plasma membrane of the inner mitochondrial membrane, all these, you can say, transmembrane proteins are present. And if you remember, I had also explained to you that on the inner mitochondrial membrane only, there is that one more transmembrane protein is present, known as mitochondrial pyruvate complex. Okay, MPC, which transports the pyruvate molecule to the matrix of the Krebs cycle, where the Krebs cycle continues, right? Also, the inner mitochondrial membrane, plasma membrane, contains a specific lipid known as cardiolipin. Okay, cardiolipin, which is a great neat question that demarcates the mitochondrial inner membrane from the, you can say, plasma membrane of any other normal cell type. Okay, so the inner mitochondrial membrane is very selective. Inner mitochondrial membrane is highly selective, you can say. Membrane is highly selective. It doesn't let, you can say, or highly selective, it doesn't let any random ion or molecule cross through it. Okay, so the inner mitochondrial membrane is impermeable to most small ions, including protons. Okay, that is why the protons also means the H plus that I was talking about cannot cross this inner mitochondrial membrane directly. Okay, it is so impermeable. Its lipid bilayer is so much, you can say, impermeable. One thing is cardiolipin, it has completely non-polar structure, so plus or charge structure cannot pass through. So what I showed you was that the inner mitochondrial membrane, it requires specific transporters, right? These transporters, these channels to throw out H plus, right? It requires channels to throw out H plus. So that is what it requires. So this is what happens, okay? It requires channels to throw out H plus. So directly through the membrane, H plus cannot go through the inner mitochondrial membrane. Point to be noted. That's why it requires the proteins, and in the end also when it will return through the F1, F0 ATPase. Okay, complex 1, 2, 3, 4, here 5, or F1, F0 ATPase. So it has to come through the F1, F0 ATPase, otherwise it cannot go through, it cannot cross, it cannot surpass the inner mitochondrial membrane. Okay, always know that. Okay, so it contains multiple, you can say, transmembrane proteins. Okay, and that is the essence of, you can say, inner mitochondrial membrane. Okay, we know it very well. Okay, fatty acid catabolism. Okay, then you can say citric acid metabolism pathway, beta oxidation of the fatty acid, that is the fatty acid catabolism, then amino acid oxidation enzymes. Okay, so all these are present in the matrix. Okay, and inner mitochondrial membrane, it contains the respiratory electron carriers like complex 1 to complex 4. Then also one more protein known as ADP, ATP translocase, and ATP synthase, and also other membrane transporters. So all these are present in the inner mitochondrial membrane, and the matrix, the inside of the inner mitochondrial membrane, it contains the pyruvate dehydrogenase complex that we had discussed in the Krebs cycle, then the enzymes of the Krebs cycle, citric acid cycle, enzymes TCA cycle, enzymes, then the beta oxidation of the fatty acid, means when the fatty acids are broken down, fats are broken down to give you energy, acetyl CoA. Then acetyl CoA will go through the Krebs cycle to generate all this electron carriers, and these electron carriers will give you energy. So that's how fats give you energy, right? Because fats give you acetyl CoA. Acetyl CoA is the starting point of the Krebs cycle, right? Acetyl CoA will react with, you can say, your oxaloacetate to form the citrate, and then Krebs cycle will be done. After the Krebs cycle is done, you will have a lot of reducing equivalents which are generated. Those reducing equivalents are NADH and FADH2. They will carry on electron transport chain, and then you will be able to generate the electrochemical gradient via chemosmotic theory, and then that will return through the F1, F1 ATPase, and it will produce a lot of ATP molecules. That is why fats are great sources for energy. If you eat good fats, okay, not the trans fats, you can say not the saturated one, lot of polyunsaturated fatty acids, and you exercise, then fats are good for your body. Okay, fats are not bad for your body in that regard. Okay, so that is the hallmark of the electron transport chain. Okay, so what happens is, you can say, I can write down an equation for you, that you can say the NADH, okay, what it does is, it does what? It oxidizes the the NAD plus would oxidize the substrate, and NADH and H plus would be produced, right? So that is vice versa. So the thing is, NADH plus H plus oxidized substrate, it will be a reversible reaction. What will it produce? It will produce the reduced substrate that we are looking at, right? That is complex 1 plus NAD plus. So that is known as oxidative process, then later on oxidative phosphorylation. Okay. So NAD linked, okay, the enzyme would be what here? The enzyme would be NAD linked dehydrogenases, because these oxidation reduction reactions are done by what? They are done by dehydrogenases. Okay. So the thing is, NAD linked dehydrogenases, what they do is they remove two hydrogen atoms from their substrates. Okay, know it very very carefully. Two hydrogen atoms from their substrates. Okay, one of these hydrogen atoms is transferred as a hydride ion, that is H minus ion, as a hydride ion, it is H minus ion. Okay, wait. As a hydride ion 2, you can say NAD plus. Okay, and the other is released as H plus in the medium to NAD plus, forming NADH, and the other is released as H plus in the medium. That's why we say that this is the reaction NAD plus plus reduced substrate, you get NADH plus H plus, because it takes away two hydrogen atoms. Okay, always remember, two hydrogen atoms from this its reduced substrate. Always know it, the dehydrogenase enzyme, okay, will remove two hydrogenase atoms, means this reaction is done by a dehydrogenase enzyme, NAD linked dehydrogenase, that is the oxidoreductase enzyme, right? Oxidation reductions, it will fall under the category of oxidoreductase. So that will remove two hydrogen atoms from the substrate. Always know this fact, that how NADH can cause this kind of reaction. Okay. Now, one more thing I'll tell you, that we have looked at fat, so they are also flavine mononucleotides, okay, FMN also. Those are known as flavoproteins that we saw in the Krebs cycle. Like fat, they are known as flavoproteins. Just like NADH, they are the electron carriers. So the flavoproteins, they contain a very tightly, okay, sometimes covalently bound, okay, those proteins can contain tightly. Tightly means by non-covalent interaction, or even covalently bound, means true chemical bond, FMN or FAD. Okay, FMN is nothing but flavine mononucleotide. FAD is flavine adenine dinucleotide. Okay, now what can happen is the oxidized flavine nucleotide can accept either one electron, okay, or two electrons. Okay, so it can yield you, or it can give you, by accepting electron, FAD-H2, and FMN can accept electron and it can give FMN-H2 also. So these are also great electron acceptors. Okay, just like your NADH. Okay, so the thing is, now I'll tell you how the electrons pass through a series of membrane bound carriers. Means there are multiple small molecules which are also bound, okay, to the membrane, and electron goes through the carriers. Good evening, Arya. Yeah, yes, Pratham. We'll be doing that from the next Zoom class, okay? So you can send the topics to me. I'll just explain it to you, and side by side we will take the queries also of other students also. So we'll be doing that from the next class. That's why I've kept one class in Zoom for biotechnology for proper interaction. Okay, so that all the doubts of the previous, of the current can be completely, you can say, resolved and clarified. And if you want any topic to be taught, I can teach it to you again also. So this class will be going as per the course. In the Zoom class, we will be going through the discussion portion. Okay. So what we have is electron acceptors, okay, or electron carriers. So the mitochondrial respiratory chain, it consists of a series of sequentially acting electron carriers, okay, most of which are the integral membrane proteins present on the inner mitochondrial membrane, and they do contain also the prosthetic groups capable of accepting or donating either one or two electrons. Okay, prosthetic group means inorganic groups. So I'll just make it very easy for you. There are three types of electron transfers which occur in oxidative phosphorylation. Okay, just know that three types of electron transfers. Electron transfer that occur in, you can say, oxidative phosphorylation. Okay, so number one type of electron, you can say, transfer that is occurring in the oxidative phosphorylation is direct transfer of electrons. Okay, that is known as direct transfer of electrons. Like one example is if you reduce Fe3 plus to Fe2 plus, that is known as direct transfer of electrons. Okay, direct transfer of electrons. So I'll give you an example. When we reduce Fe3 plus to Fe2 plus by transferring an electron. Number two is very interesting. Transfer of electrons or transfer of electron as a hydrogen atom. Okay, that is H plus E minus. In case of, you can say, NADH, right? Transfer of electron as a hydrogen atom also it can be done. Transfer of electron as a hydrogen atom. H atom. That is H plus plus E minus. Okay. Plus E minus. Then number three, transfer also as a hydride ion which bears two electrons. That one we saw in case of the direct case of NADH, right? Transfer as a hydride ion. Transfer as a hydride ion. Okay, this one. H minus ion. Okay, which bears two electrons. One hydride ion bears two electrons, right? That is what happens in case of NAD plus dehydrogenase. So NAD plus dehydrogenase transfers two H atom in case in the form of hydride ion. Okay, the one which I showed you here, if you remember. So I can go up. Okay, so that is what happened. You scroll up this screen. So before flavoproteins, we were discussing about this thing. See here, the 2H atoms. Okay, so this hydride ion is produced, and then what happens is, you can say, the hydride ion. So one of the complete H plus is transferred to the, or H minus is transferred to the NAD plus, making it, you can say, NADH, and H plus is thrown off. Other H plus is thrown off. So two electrons are transferred, but H plus is thrown off, right? So that is how you can say the transport happens. Okay. So two H plus ions, right? Or you can say the NAD link dehydrogenase does what? It removes two hydrogen atoms from their substrate, and one of that hydrogen atom is transferred as a hydride ion to NAD plus, and the other one, the other H plus, is released as H plus. The other hydrogen atom is released as H plus, because from that H, one extra electron is taken, is brought to the hydride ion. So you make NADH. NAD plus is converted to NADH, and other H plus is transferred as only H plus only. Okay, so that is the fundamental process there which happens. So now after this, we will be talking about, we can say, three other types of electron carrying molecules which function in the respiratory, you can say, chain or the electron transport system. Three other types, very very important for any examination. Okay, so in your school these three molecules can be directly asked because it is given in your book also. So the thing is, in addition to NAD and flavoproteins, okay, that FAD H2, FMN, NAD, there are three other types of electron carrying molecules which function in the respiratory chain. Okay, number one is known as a hydrophobic quinone molecule known as ubiquinone. So there are three other types of molecules except NAD plus, or NADH I should be saying, then FMN H2, then FAD H2, okay, which can carry electrons. So number one is what? Number one is known as a hydrophobic quinone molecule known as ubiquinone. A hydrophobic quinone known as ubiquinone. I'll just show you the structure also later on. Ubiquinone, okay. Number two are known as, you can say, iron containing proteins. Okay, there are two different kinds of iron containing proteins. Number one are known as cytochromes. Okay, cytochromes, iron containing protein. Number three is known as iron-sulphur proteins. Iron-sulphur protein. So there are three types of molecules except NAD plus, and also you can say the FMN H2 flavin mononucleotide, okay, and flavinadenine dinucleotide, okay, H2, that can carry the electrons. And why they are necessary is why I'll tell you. See, electrons cannot directly jump This is complex 1, this is complex 2, this is complex 3, this is complex 4. Okay, I'm not drawing complex 5 here. You may be thinking, why are they necessary? Okay, so these are intermembrane proteins. Electron cannot, when NADH has come and bound here, it has donated one electron to this one more NADH, donated electron to this one, it has pumped 4s plus outside. Now the electron needs to be transferred from complex 1 to complex 2. So it cannot direct jump, right? There is no jumping style, or there is no, it cannot just float and swim its way through. There needs to be a carrier of electron which can carry electron from complex 1 to complex 2. Who will do it? The player is ubiquinone, or the carrier of that electron is ubiquinone, also known as coenzyme Q. Okay, these supplements are also formed. You can say also are available. You can check on Amazon. You can check by the name ubiquinone. Okay, on means it is the, you can say, ketone group, and the reduced one, when you have added electron to this, it becomes ubiquinol. Okay, now ubiquinol will be reduced after it accepts the electron to ubiquinol, and then it will transfer the electron to complex 2 and again become ubiquinol. Okay, the thing is, this ubiquinone and ubiquinol, this ubiquinol is much better because it is a reduced form, it contains electrons. So a person who is having some heart ailment like heart failure, heart muscle, you can say weakness in layman term. So if that is what you are facing from, or you are facing from high BP, hypertension, then American Heart Association AHA recommends you to take ubiquinol supplements, since they will be they will be their presence will be more, their concentration will be more here. So more ubiquinol, okay, QH2 will be present, reduced form ubiquinol QH2 will be present, and they can carry more electrons from complex 1 to complex 2. Thereby they can accelerate ATP production, and your heart can then generate more and more ATP. Okay, so the thing is, it is a very heart healthy, you can say, supplement, very expensive, but still these are being recommended to people in US and Europe who are suffering from heart attack, hypertension, along with the medications that they are being prescribed for. Okay, so these are also one point of, you can say, pondering from the conceptual point that ubiquinol can be less in some person due to some kind of genetic deficiency, okay, due to excessive toxicity, too much binge eating and fried fruits and this and that, too, then that leads to hypertension, and heart is under pressure, heart needs to generate more ATP. Okay, then again ubiquinol shortage is happening, so you can take ubiquinol supplements in order to enhance your electron transport chain and in order to increase the ATP production in the electron transport chain. So one more thing I'll tell you about the ubiquinone, which is also known as coenzyme Q. It is lipid soluble, so it can transport the electron from, through the membrane from one carrier to the other, means from complex 1 to complex 2, because it is lipid soluble. So it can, suppose this is the thickness of the membrane, it can take electrons from here and it can deliver it here. So it is a lipid soluble molecule. So chemically it is a lipid soluble benzoquinone with a long isoprenoid side chain. Okay, benzoquinone with a long isoprenoid side chain. So its lipid solubility is of great importance. I'll just show you the structure of uh, ubiquinone, okay, or ubiquinol. Anything you can see reduced form. You can see 800 mg, sorry, 200 mg, 300 mg are there. Okay, so those supplements are available. Okay, you can see here the structure of ubiquinol and ubiquinone both. Okay, you can check this structure. Okay, this is the structure of ubiquinone. This is the structure of ubiquinol. Okay, these are the, this is the oxidized one. This is the reduced one. Okay, ubiquinol. If you see here, you can look at the structure. Okay, that is a zoomed in structure. Okay, ubiquinol to ubiquinol. Okay, so the next specificities of the Krebs cycle we'll discuss in the next biotechnology class, and the class on Saturday will be an interactive class again. Okay, so do join for that class and get your queries resolved. Okay, then if you have any queries, you can ask me on my personal number or in the Zoom class. Okay, then thanks a lot. Then let's meet you in the